In the last 5 years, regulators start to work in a more close way with the developers and with the CDMO in order to go faster in the approval of this type of products, and also in order to define specific rules and specific path for cell and gene. We have a lot of work to do in order to go in a more straightforward way, and in order to arrive to the final submission with more clear rules and more clear definitions of the regulatory expectation. But I can say that we saw and we observed a lot of improvement in these relations. And this is one thing. The other is for sure the manufacturing. We are now with processes that have a lot of manual steps, that have a lot of, not a lot of automation, and the scale are more similar to lab scale respect industrial scales. So also from a manufacturing point of view, we need to improve this, go to automation, scale up the processes. We are hoping that this field can grow with a new application that can help different set of patients. We know that rare disease is is a big category of diseases for which there is a lot of needs. A lot of children and a lot of patients are suffering, and often they don't have any alternative. They don't have any cure. Cell and gene is an alternative. These need a lot of preclinical work and research work, but we are here to support developers when they need to transfer the idea from the lab scale to the clinic. This is one of the more exciting parts. The other is finally to have this type of products as a real drug on the market. And so cell and gene now is a promise. We want to realize this promise. The main challenging part is to develop processes that have an intrinsic variability due to the starting biological materials, that is blood, apheresis, or cord blood or bone marrow, and develop processes with high level of quality and reproducibility starting from material with this so high variability is very challenging. The other challenging part is the development of processes using instruments, equipments, and materials that are not designed for this scope. And so try to find different solutions and solve problems that are specific for the development of this type of drugs. So the quality is an important part of our job. We need to develop a product with high level of quality, and found a compromise between the production of drugs that are alive drug as cells and viruses, and maintain high the level of quality and the control. For this reason, we have a lot of testing for the characterization of our products that permit us to verify that the product is safe and is ready to be infused to the patient. We have particular challenges because the more simple way to assure, for example, the sterility of the drugs is the sterilizing filtration. But we cannot apply in a very straightforward way sterilizing filtration to cells. And so our products are sterile by design. And this is a particular point of view, a particular aspect of our technology. All this path is done in collaboration between us, our clients, authorities, and also the suppliers of reagents, of instruments, and the technical material. So gene therapy is a team working job that we can do together, all the different partners that we have in this journey. What is relevant is to work as a team, finding a solution and trying to develop what we call platform. The platform permits us to plug and play and go in a more fast way from the development to the GMP and to the final production of these drugs.